and welcome to today's webinar sponsored by the HIBAR Research Alliance, also known as the HRA. I'm Scott Slovic from the University of Idaho, and I'll moderate today's one-hour event. This is the 7th in our series of webinars for researchers, research administrators, and others who are interested in highly integrative, basic, and responsive research, or HIBAR. The HIBAR Research Alliance website describes many of the activities of the organization, and recordings of all of our webinars, as well as abbreviated summaries of key takeaways, are posted at www.hibar-research.org. I'd like to encourage interested attendees to visit the website and consider joining our mailing list to receive occasional updates about important meetings and other events relevant to HIBAR research. Before our featured presentation by Dr. Maude David, I would like to explain the structure of today's webinar. We will begin with a quick overview of HIBAR research concept by Dr. Lauren Whitehead, the director of the HRA. Next, Dr. David will speak for approximately 25 minutes about her HIBAR collaboration with researchers at the Stanford Medical School and Second Genome, a San Francisco-based company, to use data from the microbiome to search for new treatments for autism spectrum disorder. Then we'll have approximately 20 minutes for Q&A, followed by an opportunity for Dr. David to briefly offer her final takeaways. So now let's get started. Dr. Lauren Whitehead is a professor in the Department of Physics and Astronomy at the University of British Columbia, and he'll offer a brief introduction to the concept of HIBAR and to the HRA before we turn to our featured presentation. Dr. Whitehead?
Well, thank you, Scott. I'm just going to share my screen here. And good day, everyone. Thanks for being here today. I'll be very brief, but I'd just like to say a few words about HIBAR research and the HIBAR Research Alliance. So HIBAR research is an idea that involves classifying research projects. So here's one way of doing that. This is a distinction where projects are, it's kind of related to the why one does research. Research projects could be shown as in purple here with an orientation for just discovery, coming up with new knowledge, or as shown in green, they could be intended to have public impact. And when both are true, which is sometimes the case, you have that overlap zone shown in gray, which many people describe as use-inspired basic research. So that's an interesting distinction or classification regarding why one might do research. And for short, we can call that basic and responsive. Another outlook is shown here, and it's more related to the who and the how. So in beige, you see a distinction which we call societal co-leadership, which is extremely important for HIBAR research. This is research that is connected with experts in society who are close to a problem of interest and can help actually partner with the research to achieve success. And in light blue, you see the classification of what could be called sustained urgency, which is recognizing that problems, important problems, are usually long-term. They take a long time to solve, and they only get solved if you can maintain urgency over that time. So that combination we could call partnered visionary research, or here we'll actually use a short form, highly integrative, to describe that aspect. Well, of course, I'm heading to this, which is what happens when you combine all four of those characteristics. Then you get what we call highly integrative, basic, and responsive research, which is in the center where I've shown this question mark. We call that HIBAR research. And the significance of it is that that research has historically been very, very productive, both in terms of creating new ideas and also new solutions for society. So the HIBAR Research Alliance is connected with this. Its mission is to try to have more HIBAR research occur by being a distributed network of experienced volunteers who understand and care about HIBAR research and who are well organized and united in their support of HIBAR research. So the goal of these individuals working together is to increase the prevalence of university HIBAR research from about 1 project in 20 at universities today, which is about the rate that it happens, to about 1 project in 5, in other words, 4 times more by 2030, while also improving all types of research. So there's nothing lost here, but there's something tremendous to be gained. we believe that this will help universities to respond more effectively to societal needs while also increasing the excellence of their fundamental research. So it's just such a pleasure to be here while we introduce Maude, because her research that she'll be describing really epitomizes these four characteristics that I've just mentioned. And I'll just briefly run through them to describe that. First of all, it's discovery oriented. The research does seek new knowledge about the connection, in this case, between the human microbiome and central nervous system disorders. It also has public impact, or will have public impact. It's solution oriented with a goal of lasting improvements for human health. It also involves, at a very deep level, societal co-leadership, as you'll learn. The work emphasizes trusted, equitable societal collaboration with non-university partners, including healthcare practitioners and their patients. And lastly, and over a long period, it has sustained urgency. It's going to achieve long-term objectives by building, and has been building and will continue to build, trust over time in an extremely sensitive and complex area of research. So I am very much looking forward to the presentation, Maude. And on that note, Scott, I will hand back to you. Thank you.
Thanks very much, Lauren. And now I'm pleased to introduce today's featured presenter, Dr. Maude David. Dr. David is an assistant professor in the Department of Microbiology at Oregon State University, where her lab studies the gut-brain axis in order to understand how microbes can impact our behavior, particularly with regard to autism spectrum disorder and anxiety. Today, she'll talk about her project conducted jointly with colleagues at Stanford and Second Genome that uses a crowdsource approach to understand microbial molecular mechanisms and search for new treatments for autism spectrum disorder. Dr. David?
Well, good morning. Thank you so much for the invitation. This is really a pleasure to be here today. So let me introduce you this project that I'd like to present today, which is a project that explores associations between the microbiome and autism. So as Scott just said, I'm an assistant professor at Oregon State University. I have a co-appointment in the departments of microbiology and in pharmaceutical sciences. I also added my email here. Please feel free to reach out after this talk if you want any follow-up or you have any questions. I have a conflict of interest slide here, as I am a partner in some companies. And today we're going to talk about autism spectrum disorder. So autism is a very complex developmental condition that involve challenges in multiple areas. So it's in social interactions, of course, but in speech and nonverbal communication as well. And it also involves restricted and repetitive behaviors. So this is a disorder that is fairly prevalent in the US. It touches about 1 out of 54 children. So that's the latest CDC prevalence survey that was done in March last year. So those are really the latest data. And it appears, very early in childhood development. It's usually diagnosed somewhere between 2 and 4 years old. It can go, sometimes takes up to 8 years old to be diagnosed. And what everyone really is working on with autism is what we call the etiology, meaning, what causes autism? And it's still very much up for debate. There is a very strong genetic component. There is a plethora of literature on that, but there is also a lot of literature that emphasizes that there are complex interactions between the genetic and the environment, right? So that's something we're going to talk more about today. So in addition to all those social and different behavioral symptoms I just mentioned, we also have an associations that has been observed in children with autism with a gastrointestinal status. So basically, a lot of this research started with a paper from Adams in 2011 that even went all the way to show that some of the status of these gastrointestinal problems were associated, correlated with the severity of the symptoms you observed. If you look into the CDC website, they report 17 to 86% of children with autism have more gastrointestinal symptoms. And that really launched a lot of the research on the gut microbiota. And the scientific community started to wonder if there was an associations between autism and the gut microbiota. And a lot of association studies started to be done where we looked specifically at the bacteria present in the stool of children with autism and compare them to control cohorts. But I'd say a paper that really was groundbreaking was in 2013, and it was in animal models. In 2013, a team at Caltech, Masmarian team, showed that if you feed this commensal bacteria to a mouse, you can improve some of the behavior testing that you would observe in the mouse. Another very interesting study that happened in the last few years was a microbial transfer study where they provided microbial, I would say, fecal transplants to children. And those children had GI symptoms. And with the transfer, the team showed it was in a state that the children were doing better in terms of their GI disorder, gastrointestinal disorder, but also some of the behavioral symptoms. So with all of this in mind, we started to work on this project, which is called the M3 project, Metabolite, Microbiome, and the Mind. And that tries to identify specifically the biomarker. So they could be bacteria or little molecule the bacteria produce, little compounds, using what we call a multi-omics approach, meaning using multiple technologies to try to understand how the gut microbiota could be associated with autism and potentially impact it. In the study I showed before, there was a lot of proof of principle, and the literature is pretty fantastic for that. But what we were really looking for is potential therapeutic targets. And the study I'm going to present today, we tried to address some of the limitation from previous study. For example, the sample size in the previous study, having matching controls. Like if you talk about the gut microbiota, there is differences in the diet, but also in the environment where the participants live. We also integrated a longitudinal data sampling instead of just looking at one-time snapshot. And finally, we try to take into account the whole spectrum of the symptoms of autism, not only a binary response. Does a person have autism or not? So this project, as was mentioned several times in the introduction, is a collaboration. It's actually funded by NIH under what's called a Small Business Innovation Research Grant. Those grants are meant to bring innovation into commercialization and to society, really. So the goal was to try to work together with the private sector. So here's Second Genome. And this is a collaboration with Stanford School of Medicine as well. And here are all the PIs involved. So at Stanford School of Medicine, there is a team of Dr. Dennis Wall that took care of quite a lot of the recruitment part. Professor Theo Palmer, who's a very renowned neuroscientist. And at Second Genome, the PI carrying the project is Dr. Shoko Iwai, supported and helped by Todd DeSantis and Karen DeBag, the CEO. So let's talk about this project a little bit more. The way we decided to design this study was to try to really find matching controls between children who have autism and the cohort we were going to compare them to. So the challenge here was to try to have children that would show differences due to some of their diagnostics and not too much due to other external factors. So here is the structure we decided to use. We looked for families that had two children, one with autism and one without. And to do that, we only looked at children that were between 2 and 7 years old, so fairly young in age, and they had to be within 2 years of each other, the two siblings. So of course, if you think about this, that seems like really complicated to go recruit this cohort in one specific place. So we decided to crowdsource this study. And we had to go and really put the word on social media and try to recruit as many families as we could. So the moment you decide to go crowdsource a study, that means really communicating with a lot of different participants and be able to express what you want and what you need for the study without having a one-on-one conversation in a specific place. So what we did is we developed a protocol and kits that we shipped out to all the participants. After screening, of course, first of all, they had a child that had a diagnosis confirmed with autism, but also that they respond to all the criteria that we wanted. So here is, for example, a snapshot, right, of the little flyers and protocol in the box that we sent to the parents. And this was mostly spearheaded by the Wall Lab, who really had a lot of work in contacting everyone and making sure they respond to any questions. So the way the study worked is we asked stool samples every 2 weeks. And we had some stool sample was preserved for sequencing. Some other were frozen from metabolomics. We also gathered saliva information and a lot of questions. We also asked for a video, and I'm going to go back to the why we asked for a video. So in total, we collected about 365 metadata features. After putting them together, we tried to assess inter and intra-family variability, and we really did this to investigate the environmental factors that could influence the microbiome here. So the first part I want to talk about today is a little bit of the results on the dietary informations and how we confirm the autism diagnosis and how we try to probe for the severity of the symptoms we could see. The second part, I'm just going to talk a little bit snapshot of the results on what we call the ribosome sequencing, and I'm going to explain a little bit what that is. And then at the end, I really would like to talk about how we try to engage with participants and all the challenges we had to face or to work with here. So let's talk a little bit of what we call the metadata, even though I don't like that term at all, because metadata are data. You know, the cell phone company takes metadata, they take data, they just really make, you know, a lot out of those data. So what we really gathered here were a lot of information about diets, especially diets, because as you're studying the microbiome, you have to pay really attention to a lot of the factors that could be associated and influence it. So to give you an example in terms of numbers, we got over 1,300 people signed up in the study, but after a lot of checking eligibility, etc., we ended up with 71 samples. And then when we filter down to only keep the family that gave us all the required sample, and we made sure that we use this tool developed by the Wall Lab, and we try to figure out if we could confirm the diagnosis of autism, and I'll go back to that. We ended up with only 462 samples. And at the end of the day, we really work with 432 samples that also passed some QC for the quality control for the sequencing. One thing that we used, it's published by the Wall Lab, is a video classifier where the children with and without autism would send us a video of the natural behavior in their house. And we have 3 raters that would answer specific questions while watching the videos. And those raters will just try to confirm if we could see or not if the child seems to have autism. The reason why we did that was not that we didn't trust anyone. It's just that because it's a very big crowdsourced study, we needed to have some idea of how we were doing in terms of reported data versus to confirmation of the diagnosis. We remove about 7%, right, of the samples where the diagnosis didn't seem to agree with our classifier. So those were removed from the final results of the study. So if we look at our results here, what we mostly found was that we had a few dietary differences between the cohort. And those differences could be a problem if they are found to be associated with changes in the gut microbiota. So for example, some of them were reported widely in the literature already. We're not the first one to report that the dairy consumption changes. Lactose intolerance is something that has been known in autism before. So we found this in our study as well. So it's also the functional bowel findings, diarrhea, constipations, that's different in the 2 cohorts. And so those are potential confounding factors because you don't know if the microbiome difference you may see are due to this specific issue with the children with autism, or if it's just associated with the phenotype of autism. So those could be some issue too. So we try to really carefully document those associations. The next thing that we did, as I mentioned, is we use this mobile autism risk assessment, which provide us a score and give us an idea of where the children were in the spectrum of autism. And I don't want to go into the detail, but this is really interesting because we did find specific association between the microbial structure. What I mean is, in this case, was what we call the alpha diversity, which is how diverse is the microbiota when compared to the severity of the symptoms we observed of that score. So it was really interesting to gather this information because we were able to confirm what was found in the literature before, which seems in association with the gut microbiota and the severity of the symptoms we were observing. So to dive a little bit more specifically on one of the results we found, I'm going to talk about a technology that is called 6-ness implicant sequencing. So it seems very complicated, but it's really not. So what I try to summarize here is, let's imagine you have all your bacteria, right, that we gather in those stool samples, and then we extract their DNA. And in their DNA, what we looked for was a specific region of the DNA that is present in the ribosome. And this is very convenient because we use it as tag to tell us which type of bacteria are present using sequencing technology. So here is an example of what you get at the end. And if you have the same color, that means that this is the same sequence and we think the same bacteria. This is widely used in microbial ecology. This is very good for tagging specific taxa. It gives you a resolution to the level of family, to the genus of the bacteria, something around there. Okay, so it just really, the take-home message here is all you need to do is to find this little tag and sequence it, and you have an idea of the taxa that is present. Okay, and so we found 117 taxa that were significantly associated with one or the other cohort. So either they were enriched in autism or they were more prevalent in the control cohort. So as you remember, we did this over time, and some of the taxa really were strikingly constantly differently enriched in one cohort or the other. And so to go a little bit into detail is how could you try to evaluate if this is random or if that's really something that's strictly different, those 117 taxa between those two cohorts. So we used what we call a machine learning approach. So we use a random forest for those who really like machine learning and have an idea. But what this does is just a technique that try to say, if I pick those taxa that I used before, how good those taxa are at distinguishing the two cohorts. And so here we found that we ended up with a evaluation of 0.66. If it was random, it would be 0.5. So that just gives you an idea, like we're definitely doing better than random when we just pick those and we try to see how we can separate, distinguish those two cohorts. But if you remember, I was really worried about all those diet habits and the diet changes that could influence the microbial community. So the next step in what we did is look at the 117 taxa and wonder, for each taxa, can we say if they are or not associated with dairy consumption or one of the dietary differences that we found? Because I try to show here, for several of them, we did find that they were impacting the microbial structure in the stool sample. So what we did is we took those 117 and we removed all the taxa that were associated with some dietary changes. And we actually obtained the same results, which tell us that the microbiota seems to bring information on its own. It's not only because it's influenced by the diet, but at least in our analysis, it may be because it's bringing more information than just the diet changes. One big drawback to this conclusion is unfortunately that maybe we just didn't detect the association, and every bacteria this association is associated with something, and that's possible. Everything is based on self-reporting. We can be completely exhaustive. I'm not going to develop this right now because that would take hours, but we did a lot of similar approaches with what we call omics techniques when we sequence the whole bacteria, when we look at metabolomics, we look at RNA, et cetera, et cetera. And we identified multiple biomarkers and we're testing several of them right now in animals. So we're looking if feeding them the compounds will make them change their behavior and improve some of the anxiety or social interactions test. The last piece of this talks is really trying to talk about the challenges and the successes we had in trying to interact with a lot of the participants and in this study. So I hope I introduced this enough, but we really had that need to crowdsource as we wanted to sample size big enough with this very particular structure for our collection of sample. And because we wanted to increase in sample size, we went crowdsourcing, but that comes with a lot of challenges. Because all of a sudden you're not tied to a single center, meaning you're going online and you're going to recruit children, and that raised a lot of red flags. So we had to work a lot with Institutional Review Board at Stanford, and they were fantastic. And we just had to put a lot of barrier for making sure the recruitment was appropriate. They had to vet on every single flyer that got out there, and of course all the consent form, et cetera, et cetera. The second challenge was a sample collection. As you saw, like I just showed you before, it is a pretty complex a study. It's longitudinal. There is multiple type of stool samples to send. Some of them are in buffer, some are frozen, some are saliva. And so we had to work on making sure that the instructions were clear. And we did a lot of testing for that. We actually asked some people in the team to try it with their children at home and to try to really optimize this as well as possible. Using self-reported data is also a challenge because there is a plethora of literature showing that there is some discrepancy between what people report and what objectively is happening. So I think confirming the diagnosis with the questionnaire-based classifier and a video classifier was a key point here. But what was important was also to use this over time, because even if one family reports something a different way than another one, you could hope that over time they're consistent with their own assessment. So if you see changes in the gut microbiota community, that should really correlate with changes in their reporting. So this is where the longitudinal data really comes into play, especially when you work with crowdsource study. The second point that I want to emphasize here is how important it was to engage the participant and their families. The dropout rates are always very high in longitudinal study, and the inclusion criteria were very drastic. As I showed at the beginning, they had to be between 2 years of each other, and the parents really had to be engaged and spend a lot of time sending us all those pre-labeled packages. So the kit design was key here, but I cannot emphasize enough the importance of the clinical coordinators. Someone has to be there almost every day answering the questions, the emails, the phone calls that parents may have concerns. You have to be here to answer concerns about, everything is very secure, but you have to transmit that information, explain how this works, and be honest with everything that will happen down the road with data, samples, anything. We also try to engage people by sending back some of the sequencing results to the participants. And so that was also something we had to work on with the institutional review board, because you do not want this to be used for any sort of diagnosis, but it's always good to raise scientific curiosity and showing the type of data we're working with. So that's what we did. And then finally, there was some press about this project, and this was wonderful. So for example, the NPI member Radio KQED came and interviewed us, and it was broadcast around San Francisco, and it was really great. That said, the first thing that happened online, of course, was a lot of misinformations in the comments about complete myth about autism, and I don't even want to relay those myths today in these presentations. And so, it's really critical, I think, that scientists occupy the space in the public communications, and I'm really, really thankful for the opportunity today, especially when it comes to things like autism that have a huge, tremendous societal impact. Misinformation when communicating science, it's just a challenge, especially right now, and it needs to be taken on by a lot of people, including scientists. That's all I wanted to present today. I will never be thankful enough to all the teams. I'm presenting this. I'm co-leading this again with Dr. Wall and Dr. Hiwai at Second Genome. I really want to thank my lab, of course, especially the analysts. Every result I show you is hours and hours of biocomputing and work and analysis and careful sequencing. So I really want to make sure I'm thanking them. And that will be it for today, for what I had to present today.
Wonderful. Thank you very much, Maude. That's really fascinating work, Maude, and obviously very important to society and highly relevant to the HIBAR model that we're talking about here. We have some questions that have already appeared in the Q&A box. And in fact, I wanted to begin by building on one of the questions that we've received specifically related to the communication challenges that you just mentioned. One of our attendees says that it sounds as though the communication challenges have been considerable, especially requiring you to share highly technical details with parents who may not have much background in microbiology. Did you have any support or training for this communication aspect of the project? And in fact, I want to know whether you have any advice for universities who may have scientists doing this kind of work in providing such training. Could you amplify the idea about the communication issue?
That's a very good question. So I guess there is really two parts to this. The first aspect is communication with the participant in this study. And we had a clinical coordinator who was Jenna Daniels at the time, and really took care of a lot of it. And Kaylee Dunlap after that. And it was their full-time job. So they were very trained on this and excellent at it. And, they really worked a lot on trying to bring the technicals to a level that was also different. A lot of parents came and they already had a absolutely amazing background on microbiota and sequencing. So I would say that it's a real challenge because you want to really answer the question of the parents and not just have a one boiled answer. So that was a fantastic work that was done by the Well Lab at Stanford. In terms of our training as scientists, Oregon State University has, where I am, workshops that you can attend to help with communications. It can be either for, it's targeted specifically for interviews or TV or different. And I think that it is really a good idea. All universities should implement that. It is key.
And related to that, Maude, in the area of kind of ethical concerns relevant to this research, I'm wondering if you could speak specifically about the ethical sensitivities related to work with children and families that require specific consideration.
Yeah, and the parents really are completely involved the moment the kid is a minor. It brings a lot of involvement here in terms of HIPAA compliance, but anyone who would share its information, any health information with any entity, it's extremely regulated in a study you're going to implement. So the fact it was with children was for us, the comfort, making sure we're not asking something too complicated. We're not asking something that will be very challenging for the parents. But in terms of security and compliance, this is all across the board, very, very structured in the US. You really have to go through the IRB, and they really look closely at Stanford. They have an entire system that's implemented to make sure that everything is secure, and even the transfer, even messages with the parents are and through secure messaging. So, yeah.
So it sounds as if you've been content with the institutional review structure as it currently exists, and feel that it's compatible with the kind of research you're trying to do.
Oh, they're very thorough. I mean, at Stanford, the IRB, every details how you're gonna receive the video, how you're gonna recruiting, every step, every flyer, every wording, every the consent form, everything is extremely protocol. It's not their first run, and that's good. This is why it needs to be centralized in the sense. These people are trained to really catch and help you set it up, and I mean, any institution at Oregon State as well. They really here as much to help you and to protect you and protect, more importantly, the participants more than anything.
Yeah, much of what you're describing, Maude, is related to the logistical challenges in conducting this kind of research and the necessity of having an institutional support structure. And one thing that attendees may be wondering is what universities could do to provide logistical support to encourage more of this kind of work. It sounds like you've had a good experience so far with this work, but do you have any broader advice about the kind of logistical support a university can provide to support not just this specific project, but similar kinds of work?
And exactly what you said, my advice is institutional support. It's like structure that individual scientists don't have to put in place on their own. You just need institutional support. You need to make sure that everything is secure. You need to be compliant. This needs to be reviewed by multiple person, several officers in the university. This needs to be institutionalized. You cannot put this on the responsibility of individual investigators. What I've seen in the last few years coming up, though, is some companies putting together structure for HIPAA compliance structure. And I think this is also great because those are audited. And that could be what the university would pick as their structure. And that seems to me, I mean, I haven't done the audit myself, obviously, but that seems something that really could be help every institutions to have a way to make sure that they can offer the support needed.
Yeah. Here's an interesting question from an attendee about what component of the project each of the multiple institutions plays in the project? For instance, did certain components of the research take place or are currently taking place at Oregon State, some at Stanford, and others at Second Genome? And how is that organized?
That's a very good question. So the Stanford was in charge of the recruitment, and they really are the one that were in contact with the team. And I worked there as a postdoc before leaving for Oregon State. So they really implemented the Wall Lab, implemented the whole crowdsourced study, so the websites, the recruitment, the sending the kits, et cetera, et cetera. So that was Stanford's part. And they also, as I showed, were responsible for the video and the classifier where we checked for confirming the diagnosis. And so that was very contained in their lab only. And that was very good because anything else after that was what we call de-identify. I've never seen a video of the kids. I've never, all the things that were next step were completely de-identify. So all we did was receive de-identify samples for sequencing, and the sequencing was done by Second Genome. So the Second Genome took care of the sequencing part. And also, this is what a lot of samples to take care of to make sure that everything is correctly labeled, that we know where we're at. It's a tremendous amount of work to sequence all the samples with the right quality, go back to the batch, do it again. This was weeks and months of work on their part. And then after that, there is the animal testing part, which is what my lab does. My lab's really in charge of taking the last final step, which also takes years to ramp up, the animal work and testing all of this. So this is how the 3-step works. The analytics part was everywhere, every time did the analytic part. We all have very talented analysts, and Second Genome had an analyst full-time on it. I had a PhD students and also working on that, and so did the Wall Lab. So for the analytics, we had the team from the 3 partners that work together, and we meet bi-weekly on those data.
Since you mentioned working with students, I wonder if you could say a word about the particular role of students in supporting this work.
Well, the students are paid by the NIH grant, right? They are completely supported. I have 2 students supported by this grant. One that works in the mouse facilities and the other one in the biocomputing area, Christine Tutter and Alex Phillips. And they both work for it. So it's major for university. These helped me cover their PhD and master program. So they're 100% completely part of this project. They drive a lot of it. I know at Stanford it was the same. And at Second Genome, they worked very closely with our students, which I think is really back to the impact of working with more projects that are impacting the society. I think this type of project is also great because it gives the students an opportunity to see what's happening in the private sector. And it's key, we all want to, I'm an academic and so are we on this call, but it's important for the students to have also a vision of what's happening outside. The student works very closely with Second Genome as well.
Thanks. Back to the partnership question that we were talking about earlier, the way the partners work. Based on your experience with this project, do you have any advice for a faculty member who's initiating a HIBAR project in terms of how to engage partners in a way that they are true partners in leading the research?
Those relationships build up on the long term. It's not something like you're going to call someone and next day you submit a grant, it gets funded. Those are, preliminary data were gathered in 2014. And I've known some of the scientists from my postdoc at Lawrence Berkeley National Lab, like in 2010, to see that this is really a long term. So that's my advice. Like early on, I would really advise to engage with stakeholders or, area that's a little bit just outside the purely academic setup, and try to engage with stakeholders. And there may be also, local companies, but they could be local station, federal agencies. Like in Corvallis, we have EPA, like a lot of local people, and try to see how your interests could work together. What are the local needs? And does that match any of your skill set or your expertise? And then I'd say the conversation starts this way, and then there's a long road to how can we make this work? What's the best route to make this work? And what's the vision everyone's having to try to also make it work? So that's my advice, like early on, take your time, go talk to, it's a little bit more difficult with the pandemic now, in a sense, to just go and have a coffee with someone who works in and in a federal agency not far from you, but it's important.
Hmm.
I also realized a lot about, I would say I came late in the autism or the neuroscience part of science really in my career, because I was a microbial ecologist for years first. And it really helps you to shape your vision of your research program and what you want to do. So it really brings you a new perspective.
Yeah, this is really helpful. Thank you. You mentioned earlier, Maude, that you are very much an academic based in academia, and we often find questions among people who are currently doing or thinking of doing HIBAR-like research about the promotion and tenure process and how this kind of work is viewed in that context. And I wonder if you could comment a little bit on how you've experienced this so far, whether doing this sort of research has posed any challenges or quite the opposite in your case.
In my case, it's been supported by my institution very much. There is a big push on innovation, especially in the College of Science and Pharmacy, and it's been very much supported. There is no questions about what societal impact this type of research has. I hope the perception of tenure and the promotions overall are evolving to really help you to, have more of a holistic view of what's happening in terms of research. And of course, we will all want very big research grant, and I don't think it's incompatible at all with it. But this type of project also brings a lot to the university. It just makes the visibility of the university, I think, is fantastic, and it's pretty recognized, I'd say, by so far by the institution, and they're very supportive and happy with it. So for me, it's been just a great experience in terms of promotion and tenure so far.
Do you think some of this holistic or broader view is filtering its way to the students in your lab so that they're not only learning about the very technical aspects of the research, but getting a bigger picture of the way this research fits into the research landscape and the way the project is structured, and also the effort to bring together both the fundamental exploration and the goal of societal impact? Are the students aware of that, and is there any formal process by which you bring them into that awareness?
I think they were, very aware by just the type of data they're working with, right? You have the answer to the question, and then they could really have a deep understanding on what we need and for information, and what the participants actually want to express. This is a total gap, and then that just showed a lot of the analysts and the students in the project how what we seem to care about, but what the general public or the participants really cared about. Like, for example, we were very clear, like, oh, we're going to be very clear. And so one of my students in my lab, Austin Martin, did a lot of the reporting to send back to the participants with some of the sequencing data. And we were like, oh, we'll be very clear, this is not diagnostics, because it is not, because this is a research project and the kits we use and everything is just a research project. But then it makes him realize that, oh, there is a huge difference in this, and why do people want those results? What are they gonna do with it? And that really, really, I think, triggered a lot of question in terms of what is the impact of what I'm doing now? Is it okay? Is it like again, back to the IRB, right? Discussion, what's okay, what is not, etc.
Speaking of impact, what about in the area of publication? Have you ever faced a situation where you had to decide between publishing a paper in a high-value journal within your specific field or taking a different path because the research impact for society might be higher with a different publication route? Or have these forms of publication always been aligned in your project?
So they've been aligned so far. We haven't had, you can go both routes, the classic publication journal, like routes just works like it should normally. And in addition to that, if we want to share more information by other media, we can do it. There is no reason for not doing both. So we haven't had a conflict so far at all on this.
Mm-hmm.
I don't really see how this could happen. Again, every data is de-identified, so really we're very safe on this one.
Mm-hmm. Mm-hmm.
We're very careful, at least. I mean.
Yeah, if we could return briefly to the question of training students, I have a question here about what kind of specific skills you would like to see in graduate students when they start working with you that they might not be receiving at earlier levels in their training as undergraduates, for instance? What kind of skills might undergraduate science students who are heading into your line of research need to acquire, including awareness of the logistics of research, skills in communication, notions about scientific ethics, and other aspects of the work that go beyond the specific technical training?
It's really Be Curious Tech Initiative that because of all the compliance, we have a lot of barrier, which is really safe for students to take initiative and propose new ideas. Just don't be scared. Don't just wait for me to tell you what to do. This is really the worst thing for a PI. I really want students to take initiative and try to propose new ideas, new approaches, be curious. Don't just execute what's said and stop there and wait. That's the worst. And be present. The other thing is, at least, in my lab, it's really teamwork. And there is nothing worse but when we ask a question and it takes 3 days for a person to answer the whole group. That really breaks the dynamic. So you really want a vibrant people that are excited and answer and are engaged. So that really, to me, are what I'm looking a lot on students, is people that are curious, that take some initiative and are engaged with other and are excited about the research.
So it sounds like that's the culture of your lab, that is highly collaborative and interactive.
Yeah, that's true. So I guess the answer is very biased toward the culture of my lab, but it's going to be kind of the case, I'm afraid, for every PI looking for students. But I'd say in this project, it's especially relevant, right? Because we're a team of 3 different entities. So if you're not responding or if you're not engaged, the communication became really complicated.
So Maude, we're nearing the end of our Q&A period, but I wonder if I could hand you a very broad question at the end. What was the biggest barrier you faced in doing this project that you didn't really expect? Or was there such a barrier that surprised you?
Not really, to be honest, because we had, as I said, a preliminary run, and I was, in the implementation of it. So we didn't have any big challenge. I'd say the barrier I always meet are, you want to gather as many information as possible without having this fatigue of asking a lot of questions to participants, right? So you can ask so many questions until the participants just answer yes, yes, yes everywhere because they're tired of answering 3 pages.
Mm-hmm. Mm-hmm.
And that is always something that surprises me. How do you define this? How do you manage to decide? There is a fine balance between the accuracy of the result and the amount of data you can gather.
To me, it seems that it's not a barrier exactly, but one issue that may have arisen has to do with conducting this work with parents involved. And the parents are obviously very concerned about their children and may be extremely eager for you to be coming up with results that will benefit their specific children, and not understanding that these discoveries take quite a bit of time. And so the impatience of the parents may be something that you run into. And how do you handle that issue?
We didn't really get that issue, no. The parents were really overall excited about the study, and we clearly sent back, the publications, it's all public accessible. And again, we sent back some of the snapshots of the sequencing, which I think really helped to keep everyone engaged. So I don't know. I don't think we had really that conversation with parents there. They were actually, they engaged. I think it's documented that the autism community is very engaged online, especially the community of participants was really willing to help and just be there. So I'd say we haven't had really that issue.
Have you and any of your research colleagues ever been asked to give a presentation like this one for the families that you're working with?
No, so I couldn't, right? Because it's all they identify. So I don't know the family at all. So that would have to be the only point of contact was the few person authorized in the lab at Stanford. So we couldn't do that, but we definitely answer every question that they had. And what they did, again, is to send out, make sure they know the publications are available. Yeah, that's how we've done this. We can't really do you know, if you do a like a seminar with all the people at once, you're kind of breaching a lot of
Interesting.
the
Of
identified
course.
criteria. Yeah, so you
Of
cannot do
course.
that.
So we did receive some very technical questions about the microbiology of this project. And I would suggest that anyone who has such questions write directly to Dr. David's email address. And I've tried to emphasize the questions more broadly related to HIBAR research. But Maude, now we have a chance for you to briefly offer a few final takeaways.
The few takeaway here, the few messages that I want to make sure go through this whole presentation are that, especially when you work on microbiota work, right? The study design is quite key. It's very important to think of your sample size and the longitudinal aspect here, for example, especially when you have self-reported information. So as I explained, is something that's important. And you know, there is a lot of factors that can affect the microbiota, which I try to emphasize, and I saw a few questions on it as well. But yes, like especially in autism, where children are picky eater, it's important to record those informations to take it in account in your interpretations of your results. I cannot emphasize enough how I believe that a scientist needs to communicate to the general public and stakeholders. It's something we really need to make sure we do, and we do a lot, and we make sure we pass the message that we want, because of a lot of misinformation online, and just because also I think it's exciting to just engage with a broader audience. And then I hope I convinced everyone in this presentation that the crowdsource was a way to do that. It was a way to engage with the general public and the participants, obviously, of this study.
Great. Well, thank you so much, Maude, for this excellent presentation. And thanks to everyone who's attended today's webinar. Please stay tuned for announcements of future events in this series.